Hey everyone, today's topic is apoptosis. So, apoptosis is defined as caspase dependent program cell death. So, apoptosis is equals to falling of leaves. So, it's called caspase dependent program cell death, controlled by genes and affects single cell or small group of cells. Now, genes associated with apoptosis are pro-apoptotic genes, anti-apoptotic genes, and sensors. Now, the pro-apoptotic genes are the Bax gene, P53 gene, most important, corticosteroids, mainly glucocorticoid, BCLX5, promote apoptosis. Pro-apoptotic genes, they promote apoptosis. Now, anti-apoptotic gene, BCL2 gene, BCLXL gene, MCL1 gene, responsible for resistance to chemotherapy drugs. Sex steroids inhibits apoptosis. And sensors, BIM, BED and Puma gene and Nexa gene, balance apoptosis. Now, revise the pro-apoptotic genes, Bax, P53 corticosteroids में glucocorticoid and BCLX5 promote apoptosis. And anti-apoptotic genes are BCL2 gene, BCLXL, MCL1. MCL1 is responsible for chemotherapy drugs and sex steroids and inhibits apoptosis. This is anti-apoptotic genes, while the sensors are PIM, BID, Puma, Nexa, and it balances apoptosis. Now, BCL2 gene associated with follicular lymphoma and MCL1 gene associated with development of resistance to chemotherapy. Now, the difference between the apoptosis and necrosis. The apoptosis affects single cells or small group of cells, while necrosis affects large group of cells. Apoptosis can be either physiological or pathological, while necrosis is always pathological. Now, the physiological apoptosis is seen in first embryogenesis. Example, removal of tail cells present in developing fetus, separation of fingers due to death of cells present between fingers if apoptosis fails to occur. Fingers will not separate, resulting in a condition known as syndactyly. Second, females of reproductive age group, menstrual cycle, the physiological apoptosis is seen. Self-reactive B and T cells, and if there is infection, then the TLC count is supposed to increase to 15,000. Suppose. So when infection ended, TLC will come back to normal, for example, to 5,000. The left increased TLC during infection undergo apoptosis. Now, the pathological apoptosis is seen in duct obstruction, salivary glands, pancreas and kidney, viral infection of hepatocytes, where we can see Councilman bodies or apoptotic body, and transplant rejection, graft versus host disease. Fourth, retinitis pigmentosa, rods responsible for dim light, rods mean wrath, and cones responsible for daylight vision, cones mean color. Excessive apoptosis of rods pigmentation leads to the night blindness. So, excessive apoptosis of rods pigmentation, night blindness occur. Endoplasmic reticulum, stress caused by deposition of misfolded proteins. Example in neurodegenerative disorders. Now, apoptosis is having two pathways, intrinsic or mitochondrial pathway and extrinsic pathway. Now, firstly, let's learn the intrinsic path or mitochondrial pathway. So, normally, we are having the BCL2 gene. So, and growth factors. BCL2 gene stops cytochrome C from leaking. As cytochrome C leakage led to the apoptosis, but if growth factor decreases, BCL2 also decreases. So, if there is decrease in growth factor, then BCL2, which is anti-apoptotic gene, so that will decrease, and because of that, the BAC and BAX increases, which will increase the apoptosis. This is in the case of growth factor deficiency. Now, because in deficiency of growth factor, there is more expression of BAC and BAX gene, then increase apoptosis. This is the intrinsic pathway of apoptosis. Here, the decreased growth factor, there will be the decreased BCL2 activation, BCL2 replaced by BAX gene. increase mitochondrial permeability and increase cytochrome C in cytoplasm. Cytochrome C leaks, increase activation of APAF, apoptosis activating factor, increase activation of caspase 9. So the intrinsic pathway का initiator caspase is caspase 9. Increase activation of proteases and endonucleases, proteases which destroy the protein and endonucleases which cut the DNA, and there will be the cell death. Now let's learn about the extrinsic pathway of apoptosis. So this is cell membrane, and this FAS or TNF receptor, and this is FADD, means FAS associated death domain, and this blue color molecule is the FAS ligand. So here the caspase 8 is present in animals while caspase 10 is present in human beings. Now the pathway is FAS ligand or TN alpha release in severe damage. FAS ligand bind to FAS and TNF receptors. Trimerization occur. Activation of FADD, which is fast associated death domain. Pro-caspase 8 get converted into caspase 8. Increase activation of caspase 3, 6, 7. Increase activation of proteases and endonucleases, and there will be the cell death. Now the caspases is cysteine containing proteases acting at aspartic residues. Intrinsic pathway, initiator caspase is caspase 9. In 9 we can see IN, which means intrinsic, and executional caspases are 3, 6, 7. While in extrinsic pathway, initiator caspase is 8. E means extrinsic. Now executional caspase is 3, 6, 7. Salient features of apoptosis are: first, the cell size decreases, shrinkage occurs due to structural protein destruction by proteases. One of the earliest changes, cell size shrinkage. Second is no inflammation of cell membrane is intact in apoptosis because there is no phospholipase activation. So in apoptosis there is no inflammation because cell membrane remains intact and there is no phospholipase activation. Third is chromatin condensation due to endonuclease activation which cut the DNA. Hallmark feature and the most characteristic feature of apoptosis is chromatin condensation. So chromatin condensation is hallmark feature of apoptosis. Here in the diagram we can see this DNA. When apoptosis occur, DNA got cut into different fragments. This because of the activation of endonuclease. Now the gel electrophoresis, normal nucleic acid appear as single band, while the necrotic nucleic acid appear as smooth pattern and the apoptotic nucleic acid appear as step-ladder pattern. Now the tunnel technique used for diagnosis of apoptosis. This technique uses DUTP dye which attaches at 3OH. More amount of dye is attached in apoptosis as more fragments of DNA. More intensified color, means brown color, can be picked up by light microscope. Tunnel technique means TDT, terminal deoxynucleotide transferase, DUTP, N means Nick, E and L labeling. Staining is done by annexin 5. Flipping concept is there. Now the increased flipped molecule, it means attracts more phagocytic increases phagocytic cells. No time for enzyme leakage. Cell membrane intact, means no inflammation. Annexin 5 attaches to flipped molecules, indicates apoptosis in cell is about to start. Means if annexin 5 attached to flipped molecules, it will indicate that the apoptosis is about to start. Molecule that can flip over are expression increases at the time of apoptosis. Phosphatidylserine C1q, thrombospondin. Now some important points for exam are MCL1 gene associated with development of resistance to chemotherapy, In traumatic neuronal injury, AIF, apoptosis initiator factor is secreted. No caspase activation directly activates endonucleases and proteases. In traumatic neuronal injury, and diaminophenyl indole DAPI, dye useful for detection of apoptosis. Now, the clinical significance of apoptosis is excessive apoptosis seen in neurodegenerative disorders and viral infections. And the reduced apoptosis is seen in autoimmune disorders. Now, P53 gene, which is protective gene, also known as caretaker, guardian of genome, pro-apoptotic gene. Very names of this P53, they are very names. Now, preventing accumulation of altered cell, activates during DNA damage. So P53 activates during DNA damage. Because there's damaged DNA, the P53 is released, which increases the apoptosis, causes death of cells containing altered DNA. Location on chromosome 17p. Mutation in P53 gene decrease apoptosis, cancers frequently increased. Around 50% of human cancers are due to P53 mutation. Commonest genetic mutation associated with cancer is P53Q gene mutation.